So let me start talking about this great man. I'm going to introduce you to this great man. He's not going to walk in because he's dead. His name is Gregor Mendel. We call him the father of genetics. Unfortunately, he died not knowing that. He died completely misunderstood. He single-handedly put a whole field of science together called genetics. It's actually named Mendelian Genetics after his name. He wrote a book, and he was one of the first people that also included math in the middle of biology. And we all know once you put math in the middle of something, no one gets it, right? And sure enough, no one understood what he was talking about, because he was easily 60 years ahead of his time, maybe even more than that. He started talking about things like DNA before we knew anything about DNA. He started talking about things about meiosis before we discovered meiosis. Now, he didn't say meiosis, he didn't say DNA or whatever. He said something happens that does this and that, because we didn't know anything about that. So, but if you open his book and you read about his work, you realize, oh, he's talking about meiosis 60 years before we discovered meiosis and all that. He's talking about DNA. That's maybe over almost 100 years later we discovered that one and figured out the molecular structure of DNA and all that. The guy was ahead of his time. I call him clever, but I also call him lucky. Yeah, now why do I call him my hero? He's my hero, besides my dad, of course. It's because it came a time in my life where I had to make a decision, and for my career, I was just like you. Should I stick it out with biology, or should I jump ship and go somewhere else, do something else with my life? So I decided I'll take one more class, and then I'll make a decision at the end of that class. And that class, thank God, happened to be genetics. I loved genetics. I loved genetics so much. I didn't hear what you said, but I said it. I loved genetics so much that I went ahead and got a master's degree in genetics and a PhD in genetics. That's how much I appreciate this guy. He's amazing what he did. So he started talking about, actually he used plants to do his work. He was actually a monk. He lived in the monastery, and they had a greenhouse on the property. And in his spare time, he used to go there and play around with these plants there. So he had a shelving unit of plants with purple flowers. He had another shelving unit of plants with white flowers. And every time he bred the purple flower with a purple flower, he got purple flowers. Every time he did that with the white flowers, white flower with white flower, he got white flowers. Why? Because he was using what's called true breeders. When you breed a purple flower plant with another purple flower plant and you always get purple flowers, that's a true breeder. And when you do that with the white flowers, then that's a true breeder too. So he went into the greenhouse one day and he said, okay, now I wasn't there, I'm just making a story there, right? Okay, I'm gonna go crazy with it today. I'm going to take a purple one and cross-pollinate it with a white one and see what happens. His prediction was that he was going to get some purple ones and some white ones. Surprise! He did not. He got only purple ones. So he bred the purple with the white, and he only got purple. Look at the difference between Gregor Mendel and I. If I had done this, I would have called myself a failed scientist. I would have jumped ship and went somewhere else and worked on something else completely, because I thought I was going to get purple and white, but I didn't. Oh well, I must be wrong. Let me do something else with my life. That's not what he did. He said there's something going on here. I was expecting purple and white. How come I only got purple? Then he started thinking about it. He said, well, wait a minute. These are plants. There's no taboo for crossing brother-sister plants. Now, humans, nothing. All right. Crossing brother-sister plants, I can do that. So he took two of these, a female and a male, and cross-pollinated them. And that's when he got the purple and the white. That's his cleverness. Again, for me, I would have stopped here and went and did something else with my life. But he didn't. He continued. He took two of these and he bred them together. And that's when he got the purple and the white. So there's something going on. Terminology. The two original ones are called the parental generation, the parents. These are like mom and dad for you. You are your parents' child. You're the F1 generation. You are your parents' F1. I am my mom and dad's F1, but I have children. My children are their F2, but they're my F1. Are we catching that? See, I would say, that's my son. But my dad would say, that's my grandson. So F2 is a fancy way of saying grandchildren of these. So F1 is the offspring of this. Another word for offspring that you will see when you're doing genetic problems is the word progeny. Progeny, offspring, children, same thing. But when you take two of these and you breed them together and you get this, now these are the F2 generation of the parental generation, or you can say the F1 of this one here. So these are his actual numbers from his notebook. Gregor Mendel counted 929 plants. If you add these two, it is 929 plants, am I right? Yeah. And out of 929, he got 705 purple flowers and 224 white ones. So he decided, maybe I'm onto something here. But let's make sure that it's not just a fluke there. So he decided to do other things. He decided to follow other characters. What does that mean? Watch. A character is like flower color, flower position, seed color, your eye color, for example. That's a character. But the different colors are the traits. For example, the character is flower color, but the traits are purple versus white. The character is flower position, but the traits is where they're positioned, axially or terminally. Seed color, yellow versus green. Seed shape, round versus wrinkled, and so forth. So he said, you know, if I'm finding something here, then I should be able to reproduce it. Because I got a 3-to-1 ratio. In the F2 generation, I got a 3-to-1 ratio. Well, if I'm onto something, I should be able to reproduce it when I look at other characters. And that's exactly what he did. These are, again, these are his numbers from his notebook. When he did the flower position, he got 651 axial versus 207 terminal. That's a 3-to-1 ratio. Seed color, yellow versus green, 3-to-1 ratio. 3-to-1 ratio, 3-to-1, 3-to-1, 3-to-1. It's all about numbers here. In genetics, you can't count 3 plants and call it a day. No, no. The more you count, the better your numbers. So look at the numbers that he's counting. Look at that, 6,000, 2,000. So he's counting like 8,000 of them to get this number. This number will be more powerful, more meaningful. The more you count, the more meaningful this number is. I mean, imagine if you just counted 2. And then you ended up having 1 yellow and 1 green. It'll be like a 1-to-1 ratio. But it's not 1-to-1 because you only counted 2. The more you count, the more meaningful this number is. That's genetics. It's all about numbers. In genetics, it's all about statistics as well. Probabilities and so forth. Again, that's where the math came in. And again, when math comes into something, people start getting afraid of it and they don't even want to look at it. And sure enough, They did not look at his work for over 60 years. They ignored his work because they had no clue what this guy was talking about. Until the discovery of meiosis. The people that put meiosis together, they described it, and so then he's like, wait a minute. I remember somebody wrote something about this. Who was that guy again? That guy, that monk, Gregor Mendel. Oh yeah, let me see his book now. Let's take his book down and read it. Oh, now it makes sense. It only made sense when they started discovering meiosis and the DNA and genes, and now it really made sense. So we had to pay respect to the guy. So we backtracked and we called him the father of genetics. And we also named the whole field Mendelian genetics to give him respect. So Mendel, Gregor Mendel said this, you know what? I think I'm on to something. But the problem now is I have to explain it to people. So he put together what is called today Mendelian Laws. I mean, look at that. He put together laws that for me to get 3 to 1, this is what must happen. See, remember all his ratios were 3 to 1, right? He said, for me to get 3 to 1 predictably there, this is what must happen for that. So he put it together and we now call them Mendelian Laws. Listen to me. He put them together a long time ago. We did not change them in any way, shape, or form. They still stand the way he put them together. Again, this guy is amazing. He cleverly said, you know what? There would not be a genetics question unless we're following alleles. Alleles are alternative versions of a gene. For example, in the human population, we have the eye color gene, but there are different kinds of eye color genes. There's your blue, your green, your hazel, your brown. Eye color gene is the character. Eye color is the character, but the traits are the different colors. So in the human population, there are different kinds of eye color genes, and that's why we have different eye colors. So what he says is this: we would not have a genetics concern if we did not have alleles. Let me give you an example. If everybody's eye color, I'm talking about everybody in the whole world, eye color was blue, would you come and ask me, hey, Dr. Saad, what do you think my child's eye color would be? I would look at you and like, blue? And hopefully you would not be shocked by that. So he says, why would we have a problem with genetics here if we only have one kind of something? So if all the eye colors were blue, then you would not have a genetics problem here. So the only time when we start talking about genetics is when we have genes with different versions. Blue, green, hazel, whatever, tall, short, everything about us is alternative versions of genes here in the human population. And now we have a genetics concern. Why was he able to produce purple versus white flowers? Because the flower color had 2 alleles, purple and white. If they were all purple, then he would not have a genetics problem. And then he realized, or we realized later, that these alleles or genes are located on chromosomes. The location of the gene on a given chromosome is called the gene's locus. Location, locus. So this could be mom's chromosome 1 and dad's chromosome 1. They both have flower color genes, but this one just happens to have the purple variety, and this one happens to have the white variety. So it'll be similar to you. You will have eye color genes on both of your chromosomes at the same location, but they could be the same variety or they could be different varieties. So a plant can have 2 purple ones, or 2 white ones, or a purple one and a white one. All the different possibilities. Okay, so we call that Mendel's first law, there must be alleles to follow. Good. Mendel's second law states that each individual has 2 copies of each gene. That is true for the most part. Listen to the exception, please. All the women in this room have 2 copies of each gene on all of your chromosomes. How many chromosome pairs do we have again? 23. Listen to the terminology. The first 22 chromosomes, chromosome 1 through 22, are collectively called autosomal chromosomes. The last chromosome, chromosome 23, is referred to as the sex chromosome. That's what determines your gender and other things. It has other genes on there. The sex chromosome for females is XX. You have 2 X chromosomes. And the genes on the X chromosome is the same as the genes on your other X chromosome. That's why I can stand here and say, females in this room have 2 copies of each gene on all of your chromosomes. The autosomal ones and the sex chromosomes, because you have 2 X chromosomes. Now, men in the room, I cannot say the same thing. For men in the room, the first 22 chromosomes, they're autosomal. You have 2 copies of the gene on each one. That's fine. It's the sex chromosome. For men, it's XY. And the genes on the X chromosome are not the same as the genes on the Y chromosome. So whatever gene it is on the X chromosome, you only have 1 of. Whatever gene that is on the Y chromosome, you only have 1 of. So we call men hemizygous for the sex chromosome. Hemizygous. Because men have 1 X chromosome and 1 Y chromosome. But all the other autosomal chromosomes, you will have 2 copies of each gene. There's always exceptions to the rule, and this is the exception here. Men are hemizygous for the sex chromosome because you have X and Y. Women, however, are XX, so you will have 2 copies of each gene on the X chromosome for women, but we'll see what we do with those later. Hang in there. Okay, the third law says that one of the 2 alleles must behave dominantly over the other one. One allele is dominant over the recessive. So what are we trying to do? We're trying to give an explanation as to why he got the 3-to-1 ratio all the time, predictably. The criteria number 1, I must be following alleles. Otherwise, I don't have a genetics problem. Criteria number 2, I must have 2 copies of each gene. Number 3, one of the alleles has to behave dominantly over the other. Meaning, if I'm here, just play along. If I'm here, you have nothing to say. I mean, you could want to say something, but you can't. I'm here, you can't talk. I'm the dominant allele over her. If I'm here, I turn off her gene expression. That's what I'm trying to say. I'm not trying to be rude to her, right? And that's what the dominant allele is going to do. And the dominant allele is always designated with a capital letter. So let's say capital P is purple, lowercase p is white. As long as you have the capital P, the lowercase p has nothing to say because it's recessive. Okay? So if she wants to say something, what do you have to do to me? Kick me out, right? I can't be here. For her to say something, I have to not be here. And that's what's coming in the next slide. So one allele has to be dominant over the other. And how does it do that? It turns off the expression of the recessive allele. All right, the next thing is, again, this is way ahead of meiosis, and he's talking, this is meiosis right here. Those alleles have to segregate during gamete formation. That's meiosis. Again, this is Mendel writing this before meiosis was discovered. He's saying they have to do this. They have to segregate into separate haploids. And it's called the Law of Segregation. Yep.
Can something be dominant caused by Down syndrome?
Say again?
Does it be like, okay, like, example, like with the father and the mother, father had wear glasses and then have a kid as a girl, but she was just because it was the dad or the mother?
Oh, that's sex-linked stuff?
Yeah.
That's next chapter. Hang in there. Keep that question. Keep that question.
It's gonna go away.
It's never gonna go away, right? Hang in there. Next chapter. So let's talk about this here. Look, this is Gregor Mendel's experiment. He took purple flowers, bred them with white flowers. Look at this here. Capital P flowers. That means the purple is the dominant trait. Lowercase p, that means the white is the recessive trait. You want me to prove that? Here's how I'm gonna prove that. Look at this flower. It's capital P, lowercase p. Watch this. As long as I'm here, you have nothing to say. Are we catching that? Why is this flower purple even though it has one copy of lowercase p? Because the capital case P is dominant over the lowercase p. As long as I'm here, you have nothing to say. That means capital P, lowercase p. That's why this flower was purple. That's why he couldn't see the white flower. Because the white flower allele is recessive. So this flower produced sperm that only had capital P. This flower produced eggs that only had lowercase p. So when the sperm, capital P, fertilized the egg, lowercase p, all of the children came out like this. But the capital P is dominant over the lowercase p. That's why they all came out purple. So look at the term, listen to the terminology, please. The letter designations are the genotypes. Which genes does this plant have? The capital P. That's the genotype. That's the genotype. That's the genotype. When the genotype has two letters that are the same, they're called homozygous. So this would be homozygous dominant. What would this be? Homozygous recessive. This is the heterozygous, the one that has one of each. So you have to know the words. Every field in science, every field has its own terminology. If you take economics, they have their own words. If you take accounting, they have their own words. And I'm still trying to figure out debit versus credit until now because I took a little bit of it, right? It's confusing. All right. If you take biology versus microbiology versus anatomy versus genetics, each of the biologies have their own words. You have to introduce yourself to the words to make sense of the information. Homozygous dominant, homozygous recessive. This is the heterozygous. What am I talking about? I'm talking about their genes, their genotypes. Now watch this. The color is the phenotype, the physical characteristic. The purple versus the white are the plant's phenotype. So Dr. Punnett put this approach together and said, you know what? I can show you a real cool way of figuring out what the genotype of the children will be, and so forth. Just give me what the genotype of the parents are, and I'll tell you how to figure out the genotype of the children and the phenotypes. You just set up what's called the Punnett Square. So you do this. Remember, Mendel took two of these and bred them together. So the male had capital P, lowercase p, and he remembered law of segregation. During meiosis, this capital P will go into a sperm, and this lowercase p will go into a separate sperm. Well, the same thing is going to happen to the female. The female was capital P, lowercase p as well. But for her, a capital P is going to enter one of the eggs, and the lowercase p is going to segregate into the other egg. So that's how we set it up. All the varieties of the sperm and the varieties of the eggs, and then we say this, watch this. If this sperm fertilizes this egg, what child do we get? Capital P, capital P. If this sperm fertilizes this egg, what child do we get? Capital P, lowercase p, and so forth, and so forth. So this is the genotype. Then we tally things up to get what is called the genotypic ratio. 1 capital P, how many of these do we have?
3.
No, not these.
2.
2, and this one? 1. So it's a 1 to 2 to 1. That's the genotypic ratio. 1 capital P to 2 capital P, lowercase p, to 1 lowercase p, lowercase p. Let me show you how to say it a different way. 1 out of 4. So 1/4 capital P. 2 out of 4. 2 over 4 is the same as a half. So a half of them are this. What would this one be? 1 out of 4. So if I say 1 to 2 to 1, it's the same thing as saying 1/4, 1/2, 1/4. Watch this one. I can also say 0.25. 0.25. What would this one be? 0.5. I can also say it this way. 25%. What would this be? 50%. What would this be? 25. What I'm trying to really say, if you're paying attention to me, is that on the exam, it can come at you in any different way. I can say 1 to 2 to 1. I can say 1/4 to 2/4 to 1/4. I can say 0.25 to 0.5 to 0.25. 25%, 50%, 25. I'm actually saying the same thing. Mathematically speaking, it's the same thing. Now, when we tally up the phenotypes, look what we end up with. 3 purple to 1 white. So look what happens here. What Mendel finally said is this. For me to get a 3 to 1 ratio, all 4 laws must be obeyed. The first law is the presence of alleles. The second law is each individual has 2 copies of each gene. Third law, 1 allele is dominant over the other, the recessive one. The fourth law is the segregation. There's actually a 5th law too. But for this one, you're dealing with 4 laws. And the other thing is this. To get the 3 to 1 ratio, you have to breed 2 of these together. Capital P, lowercase p, with capital P, lowercase p. That will always give you 3 to 1, assuming the laws are obeyed. So these are criteria. If you obey the laws, if the character obeys the laws, and you breed 2 of these together, you will always get 3 to 1. Okay? What if you breed capital P, lowercase p, with capital P, capital P? You're not going to get 3 to 1. They're still obeying the law, but you'll get different ratios. We'll try some of those. So there's a name for the cross where you're breeding 2 of these together. It's called the monohybrid cross. The only monohybrid is the one that breeds 2 of these together. If you breed a capital P or lowercase p with capital P, that is technically not a monohybrid. It's a single gene cross, but it's technically not a monohybrid. The only monohybrid is capital P, lowercase p, breeding with capital P, lowercase p. That's it. That's a monohybrid. And as long as the laws are obeyed, you will always get a 3-to-1 ratio for a monohybrid cross. So I just want to show you all the words are there. I will talk about this later. So of course he wanted to explore and add more things to his work. So he said, what if I follow 2 traits, 2 characters? That's equivalent to following blonde hair or hair color with eye color. If you're blonde, are you always blue-eyed? Not necessarily, no. But that's the whole idea here. Don't worry about the details. Listen, he's following 2 characters. And look at that. It explodes on you. Instead of 4 little tiny squares, you now have 16. So what he's following is the color, yellow versus green, versus the shape, round versus wrinkle. So here are all the possibilities. Yellow and round, green and round. Yellow, wrinkled, green, wrinkled. Those are the 4 possibilities. You got 2 characters with all 4 possibilities. Here they are. When you set up the breeding, in this case, it's called a dihybrid cross because you're dealing with 2 genes and both individuals are going to be heterozygous for both genes. So he takes 2 of these and breeds them. So he's got what, capital Y, little y, breeding with capital R, little r, crossed with, capital Y, little y, capital R, little r, crossed with capital Y, little y, capital R, little r. So 2 of these breeding together. And then he remembers that there's a 5th law. Guess what the name of that law is? You heard about it in meiosis. Independent assortment of chromosomes. In meiosis, we said, where the chromosomes end up, it's a random shuffle. They are independent of each other. And it's called independent assortment of chromosomes. Because of that 5th law, it allows me to predict all the possible sperms that can be produced from this male individual. This male individual will produce 25% of the sperm will be the variety of capital Y, capital R. Another 25% will be capital Y, little r. And another one is little y, little r, and little y, capital R. All the possible permutations. You gotta listen to this. Here's the most common mistake students make. I'll tell you something. You make one mistake here, the whole thing will be wrong. It will be completely wrong. You gotta double check your work when you do this. You will get some of these on the lab exam where you have to actually work out. You have to have a Y and an R. Which Y? Which R? I don't care. That I don't care is independent assortment of chromosomes. Are we catching that? You will get all combinations of Y and R because of independent assortment of chromosomes. Now sometimes it follows through. Sometimes it can be broken. Please listen to it. I still got 3.5 minutes with you. Well, the same thing is going to happen in the eggs. Independent assortment of chromosomes will produce all the permutations of the eggs too. Because the female genotype is also like this. So how we do it, or how I tell students to do it, is this. Capital Y, lowercase y. I'll put a slash in the lowercase y to show you that that's lowercase. This is one combination. This is another one. This is the 3rd one. This is the 4th one. Assuming all Mendelian laws are obeyed, you will get all possible combinations out of here. If you put 2 R's in this one, guess what you did? You just killed that child. Because when you put 2 R's, you're saying, you know that chromosome that had the Y? Yeah, it's not there. And that child is dead. Cannot survive that. All right? So I'm like, I'm so sorry. It's wrong. And the 10 points, they're wrong. They're gone on the exam. Please start practicing these. You must have a Y and an R. Which Y, which R, I don't care. That's independent assortment of chromosomes. Obviously, sometimes the letters are going to change, of course. But I'm saying you have to have one of these and one of these in each. Now watch this before you leave.
What?
You're going to say, listen, you're going to say, what happens if this sperm fertilizes this egg? You'll get this child. What happens if this sperm fertilizes this egg? You will get this child. And from the letter designation, you'll be able to figure out the phenotype. This is a dihybrid cross, because you're breeding two of these. You will always get 9331 all the time. Yes, sir.
I know usually